First, I want to thank everyone still staying here for the last section of the meeting, and also I want to thank the organizers to give me a chance to present my work. So I want to start with my summary of the community first. So we do the nanotechnology development for different diseases, but today I want to focus on our achievement for the central nervous system delivery for the HIV infection. Why this is very important, because like 2 years ago, NIH have an outline to say we want some safer strategy for central nervous system infection, because the CAR T system induce some concern of the neuron toxicity, and also the kick and kill strategy may induce some inflammation in the central nervous system. So there's not many choices for us to left. And the other choice, they require some delivery system to deal with this. And I hope, so today my talk can give you some ideas like what kind of strategy is available for central nervous system delivery. So from my aspect, there are 3 key requirements for the in vivo gene delivery system to achieve a successful delivery system. The first one is they need to get into the site. The second one is they need to directly get into the target cells. And the third one is they get into the cells and do the work. So in our lab, we collaborated with one lab in UCLA, Dr. Yunfeng Lu's lab, and we developed one system called the nano capsule system. And how this thing works is like this. So there are 3 components. It's very simple. The cargo is a molecule you want to deliver in, and we also have 2 components, which is monomer and cross-linker. So there are only 2 steps to synthesize this nanocapsule. The first step is the monomer and cross-linker will enrich on the surface of the molecules, and the second step is we induce in situ polymerization. So on the surface of the molecular, they will form a protein chain by the monomers. Then the cross-linker will hold all the chains together to hold them tight and make a cocoon structure. This structure is very important because it can prevent the cargo from the environment, such as the protease, nucleases, and also the heat denatures. And also, this is very stable. When I say very stable, it's like this. So we can scale up to very high amount for like non-human primate test. But if we want to transfer those materials to other sites, then we need to freeze dry them, and they are very stable in room temperature for up months to years. And this is what they look like. And you probably want to ask why it's nanocapsule, because it's more like a capsule to encapsulating everything, the cargo inside. And the size for them is very small. It's around 30 nanometers. So it's nano size. And this is also a very universal technology for different macromolecules, from protein to DNA, siRNA. The DNA is like a small fragment DNA. It's not like a chunk big DNA yet. And then RNA and also ribonuclease protein, which is available for later gene editing use. And then how we can use this technology to address the questions I just said. So the first requirement is, can we use this technology to improve the delivery to the hiding site, which the latent cells are hiding, latent virus are hiding. So we developed one nanocapsule with very special monomers, and this nanocapsule can carry positive charge and negative charge alternatively on the surface. You probably don't know why this is very important, because this thing can mask the surface charge of the nanocapsule, and then the immune response and other like clearance system won't recognize it. That's why this nanocapsule showing very long and uniform distribution inside of the in vivo. So this is a demonstration from the work we just published last month. And compared to the antibody itself, well, even after encapsulation, it's showing still a very uniform distribution. But compared to the native one, we can improve the maintenance up to 3 or 4-fold. And then another important thing is the nanocapsule can mask the immunogenicity. So we test after 3 doses continually inject, the mouse still has no response to, it still don't have any immunogenicity response. And also, we already test the nanocapsule from mice to non-human primate. They don't show any neuron toxicity, and they don't show any liver and kidney toxicity either. And then come to the most important part is why the brain delivery is so hard. So this is the antibody. Antibody is a macromolecular. So because of the blood-brain barrier block, so almost 100% of macromolecular, they cannot get into the brain. And almost 95% of the small molecular drugs, they cannot get to the brain. So that's why the native antibody, they can show very good efficiency, therapeutic efficiency in the body, but not in the brain. And with our nanocapsule, we can showing a big improvement of these deliveries into the brain. And this is only showing the delivery. You probably want to ask whether, how about the function? Can they play some functionality? So we apply the nanocapsule into very different applications in different diseases. And this is just a demonstration for the nanocapsule deliver broad neutralizing antibody in non-human primate. This is a collaboration with Dr. Nancy Hagewood. So this figure is from historic data. So the axis is the viral load in central neuron fluid. So it's kind of like an indicator of the brain infection. And the y-axis is the viral load in the blood. So the red one is untreated animal. We can see once they get a higher blood viral load, they will have higher viral load in CSF2. And however, when they treat all the monkeys with different kinds of methods, small molecular drug or antibody or like combination, we don't see any improvement in the central neuron system. So, so how to read this is, although the treatment probably can decrease the viral load in the blood, but the viral load inside the CSF is continually correlated with the viral load in the plasma. So it's more like a free diffusion viral load. So there's no specific or there's no improvement in the treatment in the central neuron system. And compared to the historic data, the blue dots are from the group treated with nanocapsule. So compared to the red one, so the central neuron system, the viral load in the central neuron system showing significant improvement of the viral load suppression. And also, so that's a little summary. We can use nanocapsule to deliver something from like protein and other things into the deep tissue, especially in the CNSF, the central neuron system. And the first requirement, we can say check. And the second one is whether we can let the things go to the target cells. And for the material science, it's very simple strategy. We just conjugate something and let them go. So this is the way we are doing. We just conjugate one target ligand on the nanocapsule. And how this thing works, I'll explain one one system we used. And so what we are using is we encapsulate rituximab, which is antibody against the CD20, and it's well used for clinical to treat B-cell lymphoma. And we conjugate one target ligand, which called CXCR13, which can bind to CXCR5, which is a receptor on B cells, which can target to the lymphoma cell, B-cell lymphoma cells. And the cells we're using, the whole system we're using is like this. Ideally, we want this CXCR13 to bind the receptor on the B cells. And so this binding will concentrate all the particles around those B cells. Then the antibody will be released out from the null capsule. And this antibody will bind to the CD20 receptor. And this binding will induce a B-cell apoptosis. The cell will die. And then the next question is whether this is very specific, how is the specificity, whether this binding is very, whether there's other non-specific binding. So this is a demonstration in vitro where we co-culture B cells and T cells, and then we add an null capsule. This is without ligands, and this is with the ligands on it. And we can see after washing out, if there's no ligands, null capsule won't stay, and they all be washed out. And however, compared to this one with the target ligands, we can see the null capsule very specific binding to the B cell, not T cells. So we use the cell line to demonstrate this is very specific binding, and then we also demonstrate whether this binding can improve the therapeutic effect in the mice or not. So this mouse model is a B-cell lymphoma model, use the same cell line just used before, and this mouse model can have systemically lymphoma, but they also can have a brain metastasis. And this is also based on humanized mice. So the humanized mice system can support the antibody function very well, including ADCC and CDC and also apoptosis. So compared to the native treated group, you can see the antibody play very good function in the body part, but not the brain. So all of them, all the mice in this group, they have very heavy tumor burden inside of the brain because of the limitation of the delivery into the brain system. And compared to the native one, the null capsule treated group, they showed clearance of the tumors. Even the signal from the brain, they can clear out later. So I hope this can convince you this is a good strategy to deliver the important macromolecules into the central nervous system. So this second requirement check. So can we directly use that for the in vivo gene editing? The answer is no. So sorry, we cannot directly use it. The reason is, I just mentioned, it's a positive negative charge alternative chemistry on the surface. That thing prevent the clearance and prevent immunogenicity. So that's also prevent getting to the cells. Any type of cell, it's so hard to get in. So how we can address that question based on this? So we do a surface modification on this null capsule. We take out the negative charge. So the null capsule will be positive charge on the surface. And this thing is very important to get into the cells because the cell membrane, most of them is negative charge. So they will bind to the cell membrane and induce apoptosis. And later, I will use this CRISPR-Cas9 gRNA system to demonstrate gene editing. I know this is not only in the gene editing method, but this is the easy one we can use for right now. So that's a demonstration. It's just proof of concept. So the question for the gene editing for those reservoirs, the question is whether they can get into those quiescent cells. So this is a demonstration of the nanocapsules, whether they can get into quiescent primary T cells. So we generate this latent infected model. So the T cells will infect with HIV, then get into the latent stage, and we transduce the cells with the nanocapsule and start to stimulate it. So the positive signal here is showing how much cells are carrying the HIV gene, and with the nanocapsule, we gene editing around 60% of the cells. And another primary HIV primary target cells is macrophages. So we also confirmed the gene editing can also play function in the macrophages, primary macrophages. And also we're lucky to have this HIV-infected non-human primary microglia cells currently provided from Dr. Nancy Hagood. We also can show in gene editing in that primary cells, in that macrophage cells. So the third question, third requirement, we can say check. But here we answer all the questions individually. How we can put them together? It's also very straightforward. We just put them together so they can have all the good things. It's just easy to say like that. So the ideal structure is like this. So we have this nanocapsule. Outside is the one who can prevent them to induce immunogenicity and other things, and also carries target ligands. And inside will be the one who can get them into the cells. But these two layers, it's easy to say but hard to do. So this is the imaging to prove we can make that kind of nanocapsule right now. So this is a mono layer. This is the first one. They can circulate and get into the brain. And this is the second one. They can get into the cells and play great function. And this is the one we put them together. You can see this particle has 2 layers. And we hope this particle can show the properties as the other 2, and they also have shown better functionality. So that's one way, that's the proof why we cannot directly use the positive charge nanocapsule directly, because when we inject them into the body, the particle will go to liver and kidney right away. And by coating the outside layer, and the particles can uni distribute again, same as the one we used for the brain delivery. And also, we're happy to see with this nanocapsule still keep the good brain delivery system, so we can still see the good intensity in the brain. And also, we confirmed this nanocapsule can get into the cells. It takes longer time, which is reasonable because they need to take off the outside layer. So it takes some time to transduce the cells well, but still, if we wait for longer enough time, it still can play the gene editing in the infected primary microglia cells. So I come to my conclusion is nanocapsule can improve the delivery into the central nervous system, and by conjugating the target ligands, they can target the target cells. And also, if we modify the surface of a nanocapsule with a positive charge, they can get into the cells and remove the HIV proviral gene. And later, if we combine everything and make a bilayer nanocapsule, we hope we can address certain level of gene editing towards the HIV cure. And I want to thank everyone, because this is a big project. I want to thank everyone who involved this project. And thank you. I'm ready for taking any questions. And I hope my talk is worthy for you to stay. Yes.
The bilayer particles seem to target the testes in that last slide you showed. Is that true? Maybe there's some use for that that particle.
Yes.
Yeah.
So we are also repeating that because we also see that opportunity to cure other diseases. This we still need to confirm it.
Okay, I have a question. If you can go to the next slide, just advance. So did you remove the microglia from the brain? Is that what you did? So,
yeah, so what I did is I isolated. It's more like concentrate the microglia cells from the brain tissue by Perco. So it's more like gravity. So it's not like a pure microglia cells, but I kind of like concentrate it. So the purity of the population is around 60 to 80% by staining CD84, CD68. Sorry.
Okay. Is that the best marker for microglia?
CD68. So what I use is I use CD11B and CD68. Right. Both. Yes. Yeah.
All right. Thank you.
Trish in the back. This will be the last question.
I just had a really quick question. So are you looking at gene editing by flow, or did you do any, is it strictly by flow, or how did you kind of measure that? And how do you know the efficiency of your editing?
Thank you for your question. So what I did here is by flow. So we stand P24, P27 for different like HIV or HIV. And for, I don't have time to explain. For this one, actually, we transduce the macrophages with a vector, which has a reporter gene. So we can also monitor the reporter gene, like the EGIP expression, to see how much they got gene editing. And also, I need to emphasize the gRNA we used is target LTR. So once they get gene editing, they should cease the virus expression.
Thanks.
Yeah. Thank you.